Okay, so hello everyone, and a very warm welcome to our webinar in collaboration with DomainX. So this is the third webinar in the drug discovery series with DomainX, and it's been wonderful so far to have so many people join us on these very engaging sessions. So today we've got Philip Rawlins, who's group leader at DomainX, who's going to talk to us about the role of various biophysical methods in drug discovery. So we're going to hear some more about the strengths and limitations of these different techniques, and then how they can be applied with examples of relevant case studies. Whilst people are still joining, I am going to play a short introductory video in which Natalie Winfield, Executive VP of Commercial Operations at DomainX, just introduces the company. So if you bear with me, I'm going to share my screen and play that now. Thank you.
Hello, thank you all for joining this webinar. I'm Natalie Winfield, Executive Vice President of Commercial Operations at DomainX. I'm going to give a very quick introduction to DomainX before we get into the science. So for those of you who don't know much about DomainX, we're a fully integrated provider of preclinical drug discovery services. We're based in the Cambridge, UK area and currently have 2 sites. Our site at Chesford Research Park houses our chemistry and bioanalytical departments, and we opened a new site at Unity Campus for our biology department at the beginning of 2024. We have received multiple awards for our services, including most recently the prestigious King's Award for Enterprise. We have a world-class team and currently have over 100 staff. Our scientists are highly qualified and experienced, with around 90% having either a master's degree or PhD. They have a wide range of expertise across disease areas and target classes. We have a global client base and work with a range of different types of companies, including biotech and pharmaceutical companies, academic institutions, and patient foundations. We offer the full range of preclinical drug discovery services, including protein production, assay biology, medicinal, computational, and analytical chemistry. We support our clients through all the stages of drug discovery, from target validation and protein production through to lead optimization. If you would like to find out more, please do get in touch. I hope you enjoy the webinar. Thanks.
Okay, excellent. So just a quick word from myself. So if you can kindly remain muted with cameras off, that would be much appreciated. But we do also want the session to be interactive. So the chat box has been enabled, and that's so you can add your comments, questions, and thoughts into there. And then after Philip has given his presentation, I will do my best to field those to him. If there's anyone throughout the Q&A session that would rather raise your hand than use the chat box, then absolutely you can do that. At the bottom of your screen, there's a react icon, so you can use that, and then I can look visually down the list of participants just to pick anyone up that way, if that's your preference. As you heard, we are recording the session as well, and that's just so if you'd like to catch up on the content afterwards to recap, or indeed if you think a colleague who would have benefited from being online today is not, and you can share it with them, then that enables that also. So with that, I think I'll hand over to your good self now, Philip, to guide us through the webinar. Thank you.
Hey, thanks for the introduction. Okay, hopefully you can see that now.
That looks good, yeah.
Yeah, thanks. So I'm going to talk about some of the ways that we can use biophysical techniques to overcome challenges in drug discovery projects. So first of all, there's a number of different biophysical techniques available. I won't talk about all of them today, but these are the ones that are available at DomainX. So we have grating coupled interferometry, or GCI, which is analogous to SPR and enables the measurement of binding kinetics. We have spectral shift on the dianthus instrument, which is a plate-based technique. We have an older instrument called the monolith that allows us to measure microscale thermophoresis. And we have ITC, which enables us to measure binding affinities in solution. And we have a couple of different methods for measuring thermal melting temperatures of proteins, either by nano-DSF or DSF. And then we also have access at the University of Cambridge to mass spectrometry, which enables us to measure the oligomeric state of proteins. So on this first slide, I'll go through the three kind of key techniques that we use at DomainX for measuring binding affinities. The first one is grating coupled interferometry. This now enables us to measure binding kinetics, so the association and dissociation of compounds or other proteins binding to proteins or other ligands. For this technique, it doesn't require much protein, and in terms of throughput, it does vary quite a bit. That's mainly determined by the kinetics of the compounds. So for fragments, with this technique, we can measure a couple of hundred fragments overnight, assuming that they dissociate quite quickly. But with much more potent compounds that have slow dissociation kinetics, then you might be limited to as little as one compound per day if it's a very slow compound to dissociate. Sorry, I just need to move my screen. The potential disadvantage of this technique is that one of the binding partners needs to be immobilized on a biosensor chip, and not all proteins or ligands are amenable to that. So that's one potential limitation. Also, the assay sensitivity is dependent on the size of the binding partners. So the larger the molecule that you immobilize on the sensor chip, and the smaller the binding partner, then the more difficult it is to measure that binding interaction. And there's also the possibility that the immobilization itself causes some kind of interference in terms of the binding interaction. So that could be via steric hindrance, or if the binding is via an active site lysine, and lysines have been used to immobilize the protein, then that can obviously interfere in the binding. Another key technique that we use is spectral shift. So the advantage of this is that binding events are measured in solution. It doesn't enable the measurement of binding kinetics routinely, but you can measure equilibrium binding affinity. It needs quite low amounts of protein, and it's higher throughput than GCI because it's the experiment is done in 384 well microplates, so you can test a number of microplates within a day. So you can test, a lot of different compounds in one day. Potential disadvantage with this technique is that one of the binding partners needs to be fluorescently labeled, so you need to find a way of being able to do that. And if the interaction is quite far away from where the protein or molecule is labeled, then sometimes the sensitivity of that binding is reduced, and it may not be possible to measure the binding. And similar to immobilization, there is a possibility that labeling method, depending what it is, could interfere with that binding interaction. Final method is ITC. So this is kind of considered to be the gold standard method. It doesn't require any labeling or immobilization. The interactions are measured in solution. And it can produce quite a lot of information. So you can get affinity, you can get enthalpy and entropy information. Also, the stoichiometry of binding can be determined using ITC. It is quite low throughput though. So one compound could be tested every couple of hours. And also the main disadvantage with this technique is that very high quantities of protein or molecule are required. So typically, it would need around about 0.5 mega protein for a single interaction experiment. And also, in addition to that, as well as quite high amounts of protein being required, the protein and the binding partner need to be at quite high concentrations, and you can start to get into issues with solubility using this technique. So a little bit more on ITC. So as I said, it's kind of considered to be one of the gold standard biophysical methods. It basically measures the heat of binding. So when two molecules come together and bind, heat is either absorbed within the reaction or emitted, and that can be measured by the very sensitive instrument available from Melbourne, the Peak ITC. So no tags or fluorescent labeling is required, and it can be used for a wide variety of different molecular interactions, such as antibody binding, DNA or RNA binding, and protein-protein interactions. And the information you get from that is the affinity of binding, the enthalpy, entropy, and stoichiometry. And one good reason to try to use this technique is that it's believed that enthalpically driven binding events may make better starting points for a drug discovery project. So moving on to grating coupled interferometry, or GCI. So this is a technique that's analogous to surface plasmon resonance, or SPR. The difference between the two techniques is just really the technology that sits in the background of the instrument. So the actual data that you see looks almost identical between the two methods. SPR relies on using a prism close to a biosensor, so that the angle of reflection is measured, and that correlates very closely with the refractive index at the surface of the biosensor chip, which relates to the mass bound to that biosensor chip. In a similar way, GCI uses a waveguide, so instead of being reflected off the biosensor, the light is actually propagated through the biosensor, potentially giving more sensitivity with GCI than SPR. And then that signal is then compared to a reference light signal, and that produces an interference pattern that is then used to correlate to the binding events. A couple of advantages of GCI are that it can measure very fast off rates, so potentially around about 10 times faster than using SPR. And it also uses disposable microfluidics. So this is very amenable to using crude reaction mixtures, such as you might do with direct biology. And the fast off rates mean that can be used for off rate screening, again, with crude reaction mixtures. So just going through a few kind of simple case studies here. So in this case, we had run a fragment screen for structured RNA using microscale thermophoresis. From the screen, we identified several hits from that screen, and we then we used GCI in this case as an orthogonal assay, so a counter screen to the MST. And we had two forms of the structured RNA, a healthy form and a disease state RNA form. So we did concentration response curves for these fragment hits, and you can see that the affinity for these fragment hits was slightly better for the disease state RNA than it was for the healthy RNA. So potentially we've got some selective fragments for the disease state RNA, which was the aim of the project. We can also apply GCI to measuring ternary complex formation, which is a very useful parameter to try to measure for bifunctional ligand and targeted protein degradation projects. So this is where you can take a PROTAC molecule such as MZ1. In this case, we use BRD3, BD2 domain, and VHL. We looked at different immobilization densities of VHL for both the binary and the ternary complex formation, using a fixed concentration of 2 micromolar of the BRD3, which was in solution, and the VHL was immobilized. So we got affinities very close to the literature values produced, so around about 30 nanomolar for the binary binding of that PROTAC just to VHL itself. And then in the presence of BRD3 to generate the ternary complex affinity, we then got 17 nanomolar for that versus 8 nanomolar from the literature. So positive cooperativity in both cases. The cooperativity value that we got was slightly lower, but in the same kind of range as was produced in the literature. Another way that you can use GCI is to measure the affinity of antibodies, so large molecules rather than just measuring small molecules. In this case, we mobilized the antibodies via a protein A and protein G chip, and then we then flowed the antigen for these antibodies over the surface to measure the binding affinity. The reason to do this is to avoid avidity effects, because if you immobilize the antigen on the surface, you then get an avidity effect from the two binding sites on an antibody. So this way we were able to get a much better estimate of the true kind of affinity of the interaction of the antibody for the antigen. You can see in this case we tested 6 different antibodies and got quite a wide range of different affinities from about 1 nanomolar up to about 200 nanomolar. So moving on now to spectral shift, which is available from Nanotemper. So as mentioned earlier, this requires the fluorescent labeling of one of the targets of interest. And the way that the instrument works is that you measure the fluorescence in the well, and the instrument measures 2 very close emission wavelengths at 650 and 670 nanometers. And the change in the ratio between the emission fluorescence at those 2 wavelengths then gives a binding signal. So if you plot the ligand concentration against that ratio at those 2 wavelengths, you can then plot a binding curve and determine the affinity of an interaction. What we found since we had this technology installed is that it gives a much better signal to noise ratio than MST and TRIC. And it also appears to be less sensitive to aggregates from proteins. We've applied this for, again, a wide range of different molecular interactions, such as RNA and molecular glues. And I'll come on to some of those examples over the next few slides. So this first example is a couple of kind of small case studies where we've looked at spectral shift for measuring molecular glues. So in these experiments, what we've done is we've fluorescently labeled one of the proteins involved in a PPI interaction. We then apply a fixed concentration of a compound, and then we titrate in the protein that binds in that interaction. So with example 1, the red line is just a DMSO control. And for this particular interaction between the PPI, there's not normally a measurable affinity between the 2 proteins. But in the presence of a compound shown in black, you can then see that you can generate a binding curve and you get a concentration-dependent interaction between those 2 proteins. On the right is an example for a PPI interaction where there is an interaction already, so quite a weak interaction, around about 2 micromolar. And we're looking for compounds that increase that affinity between the 2 proteins. So we ran a fragment screen, and I've just shown one example from that, but we were able to find a fragment that actually enhanced that interaction more than 10-fold. So it had quite even though the fragment's a very small compound, it was still able to significantly increase the interaction of those 2 proteins. And similar to GCI, we've also applied spectral shift to measuring ternary complex formation. So this first example is the same compound as shown before for GCI. So this is measuring BRD3, BD2 interaction with VHL via a PROTAC molecule, MZ1. On the left in black is the binary complex interaction. And then on the right is the ternary complex interaction. And in the literature, the cooperativity value for that, the alpha value was around about 10 via ITC. And when we tested that, we got a similar value, around about 7.6. So very similar to the literature value. And then we also tested a different PROTAC molecule, DBET1, which is again a bromine domain PROTAC that's been designed for cereblon this time instead of VHL. And when we tested this compound, we found a negative cooperativity. So the actual affinity between binary and ternary was slightly weaker for the ternary complex. We could still measure binding, and we're convinced that this is ternary complex formation. But obviously there's some kind of steric hindrance clash. And that is supported by the literature. It's just we don't have a direct comparison with BRD3. This has been done for BRD4 in the literature. And then finally, we've also used spectral shift to develop competition assays. So where you can label a small molecule with a fluorescent dye, we've done that both using a commercially available non-selective kinase inhibitor, which is shown at the bottom, and then also with a fluorescent tracer that we've developed ourselves using a kinase inhibitor, and then in-house we've made a fluorescent analog of that. And we've been able to develop assays using both of those. So at the bottom, with the non-selective kinase inhibitor, we've applied that to 3 different kinases and got nice concentration response curves for all 3. So even the titration curve in black, although that looks like a very small signal, it's still a significant binding signal and can be used to measure binding interactions of different compounds. So we took kinase B, and you can see the concentration response curves of various competing kinase inhibitors, and the affinity values for those are shown in the table. And you can see in the table, we also did the same experiments using an FP assay, so using the same probe, and we developed an FP assay and then did the same experiments measuring the competition and IC50 of different inhibitors against the 3 different kinases. And you can see that the data matches very well between spectral shift and FP for all 3 kinases, showing that this is quite a good way of doing these competition studies. We think that in terms of the actual assay development route involved, that spectral shift is actually a lot quicker. And also it doesn't have some of the issues that FP can have, such as ligand depletion. So I'm now going to move on to a bigger case study. So the examples I've given so far are just some kind of small bits of data. We've now got a case study where we've looked at the adenosine A2A receptor using our polyper approach within DomainX, which enables us to measure or express proteins in detergent-free nanodiscs. We've then run a fragment screen with the A2A receptor, and then applied direct-to-biology chemistry to that, some of the fragment hits from that screen. So just to start, so in terms of membrane proteins, they're quite difficult to use in biophysics. There's 3 potential different approaches to this. So you can use the kind of traditional approach of using detergents to extract the membrane proteins from cells. There's a large variety of detergents available for this, but the downside to using detergents is they don't generally represent the natural environment of the membrane protein once extracted. And also some of the detergents can interfere in downstream applications such as biophysics. Another approach is to use membrane scaffold protein nanodiscs. So this requires the detergent solubilization as an initial step, and then nanodiscs are then used to keep the membrane protein intact and happy. There's a number of customizable lipid compositions that can be used. They are generally suitable for downstream applications, but it still requires that detergent extraction step, and also it requires quite a bit of optimization of the scaffold protein and lipid to get the nanodisc prepared properly. So the way that we approach this, in addition to detergent preps, is to use native nanodiscs, or what we call polymers, polymer lipid particles. So the advantage of this approach is that you maintain the natural lipid environment of the membrane protein. These polymers that we use essentially act like cookie cutters and extract the membrane proteins directly from the cell membrane. They're suitable for many downstream applications, and completely avoid using detergents. So that also helps in terms of the downstream applications. Some of these are incompatible with divalent cations, which can cause a few limitations, and some are also pH sensitive. So that's something to consider. And we've applied these polyper preps to biophysical assays. So there's a couple of examples shown here. The top example is an ion channel. So we developed a GCI assay shown here. So the data on the top right shows both a kinetic affinity determined by wave rapid and also an equilibrium affinity. And you can see both sets of data match pretty well. And then at the bottom, we've expressed an ABC drug transporter and then developed an assay using microscale thermophoresis in this case, measured a very weak binding compound, ATP, known to bind to this type of bacterial ABC transporter. And we got a KD value of about 600 micromole, which is very consistent with what's reported in the literature. So in terms of A2A receptor, so this is a GPCR. There's a lot of interest in developing antagonists for A2A receptor, both in oncology, where it can be used as an immuno-oncology target, and also in neurological disorders, where it's been there's some evidence that it may be important for Parkinson's, Alzheimer's, and Huntington's disease. And there is an approved drug that was launched back in 2019 for Parkinson's disease. So recent advances in structure and assay techniques, such as the polymer prep, means there's a lot of interest in fragment-based approaches to identify novel and selective A2A antagonists. So in terms of the workflow that we have for preparing polymer preps or preparing membrane proteins in general, so we take a two-step approach to this. The first step is a screening approach. So we've now got more than 30 polymers available for the polymer method. We usually also include a few detergents, some of the common detergents that are used for membrane protein expression. We test all these in parallel, and then we identify those where we get a reasonable expression and yield from the solubilization step. So we measure that via Western blotting. And then we have an affinity capture step, so that the tag on the membrane protein is then used to extract it from the rest of the prep. And then we retest that in a Western blot to see which of the proteins are folded properly, because if that affinity capture step doesn't work well, it suggests that the protein is potentially misfolded. So you can see that this is important because some of the methods here, or some of the different polymers, appear to solubilize the protein quite well. But after you do the affinity capture step, you can see that some of those, you can't actually see the protein on the Western blot following the affinity capture, suggesting they may not be particularly well folded. So this workflow allows us to identify the best polymers to use for a given receptor. So we did this for A2A. And then the following step after this is to then apply some protein QC experiments, just to make sure that the protein looks like good quality. So this is things like running a gel. We also do DLS, and you can see the protein in this case for both the polymer that we use and also the detergent prep is nice and monodisperse. And then we also run size exclusion chromatography. You can see we get a nice sharp peak for the detergent, and then a slightly broader peak, as we would expect for the polymer prep. So this all looks very promising. So we then took these preps, and then we tested both the detergent solubilized prep and also the polymer prep in nano-DSF using 3 different compounds for the A2A receptor. And again, this is another kind of QC step. It allows us to quite quickly and easily test whether the membrane protein is functional within that polymer prep or the detergent prep. So you can see that for both the detergent prep and the polymer prep, we got nice TM shifts for 3 different compounds, caffeine and theophylline, which are quite small and relatively weak compounds. And then we got a very big shift for the ZM compound that's a A2A receptor antagonist. And we got very similar TMs for both preps as well. So that gave us confidence then to then develop a spectral shift assay. So we labeled the receptor via the his tag in this case. And again, we then tested 4 different compounds for binding to the A2A receptor, shown on the top right here. So you can see we've got very nice concentration response curves for these compounds, with the exception of the ZM compound, the most potent compound. And the reason that this looks a lot steeper is we actually reached the tight binding limit for this compound. So that compound's very tight. It's got a binding affinity that's sub-nanomolar. But with the exception of that, when you compare the affinities that we got for these compounds in spectral shift, it compares very well with the expected literature values for these compounds. In addition, we chose caffeine and theophylline because these are quite small and relatively weak compounds. And they're basically fragment-like. So this also gave us confidence that this would be amenable to fragment screening. So that was the next step that we took. So in terms of fragment-based lead discovery approaches, so at DomainX, we have our own in-house fragment library of about 1,200 compounds. So the biophysical techniques are very amenable to fragment screening because they're very sensitive, and they tend to suffer less in terms of interference from compounds when fragments need to be tested at high concentrations. And we also have structural biology. So once you've identified fragments, it's important to identify where the fragments bound so that you can quickly progress those compounds to find improved binders. So we ran a fragment screen. In this case, we tested 960 of our fragments at 250 micromolar against the A2A receptor via spectral shift. We used theophylline as a positive control at 100 micromolar. And also DMSO controls we used to normalize the ratio. The software identifies problem compounds. So there are compounds identified from the fragments that had autofluorescence. But we also got quite a few hits. So the spots in purple shown here all appear to be hits that seem to be well behaved. So we took those forward to the next step. We actually had quite a few hits out of the 960, about 125 were identified as potential hits at 250 micromolar. So we decided to then retest those compounds at a lower concentration at 100 micromolar to eliminate any false positives or any weak binders. And we identified 32 of the best hits from that. All of these were then tested in a full concentration response. Some examples are shown here. So you can see we've got some very good ligand efficiencies and also very good affinities. So some quite close to theophylline, one of the known binders. And when you plot the ligand efficiency against the PKD for these, so you can see we've got a lot of very ligand efficient compounds from this screen in quite a nice affinity range. So now that we had these different hits, what we decided to do was then apply our direct biology platform to these hits. So direct biology is a way of rapidly synthesizing molecules in a plate-based format, and then screening crude reaction mixtures against in the particular assay of interest, so in this case, the A2A spectral shift assay. Typically, we run around about 1,000 reactions, but this was just a case study, so we haven't done that kind of number in this particular instance. But this can be applied to a variety of different assays, both cell and biophysical assays. And it's particularly applied to targeted protein degradation projects where we can link quite easily E3 ligase warheads to the ligand for the protein of interest. But we've now started to apply it to fragment expansion, both traditional and also covalent approaches. That's what I'm going to show here. So the first step to do this is first of all just to check that the reaction mixtures used for direct biology don't actually interfere with the technology. So this initial step is just using a different assay in this case, so just MZ1 binding to BR3, as I showed earlier on in the presentation. So the 4 different coupling methods that we use were tested, and you can see both for an affinity capture method using NTA capturing of the his tag, or NHS covalent labeling, we got good binding response curves in all cases, suggesting that none of these reaction mixtures actually generally interfere with the spectral shift technology. But another important thing to do is to validate those crude reaction mixtures in the actual assay of interest. So again, we did this using 50 micromolar of the reaction mixtures. So this is not at this point using any actual compound synthesis. It's just checking to make sure that the reaction mixtures used themselves don't interfere with the assay. We found that amine coupling reactions and reductive amination reactions all work quite well in the assay, but we did see some interference with the Buchwald and the Suzuki reactions, suggesting it may be a bit more challenging to use those types of reaction mixtures. But that was okay because some of the fragment hits that we obtained were suitable for the amide coupling reactions. So we then went on to do that as the next step. So we had 4 different fragment hits that were amenable to the direct-to-biology amide coupling approach. We have an in-house amine library of about 4,700 compounds. So we applied property filters. We then used the BB select selection method that was published earlier this year, and then enumerated the different potential compounds that we could make from that to come up with 148 compounds for synthesis. So that's using 4 of the fragment hits with 37 different amines. So once those reactions were completed, we used 2 different methods actually for that amine coupling reaction. So one is shown at the top half of this plate and then the other at the bottom. So the reactions are just repeated in the top and bottom of the plate. The EDC and oxymapure methods work better, so we actually focused on that for this particular case study. And we took forward all those reaction mixtures to test in the A2A assay. So shown at the top, so these cores are the 4 different fragment hits that we had identified. And you can see this top half is the how much of the reaction mixtures has converted, the estimated conversion rate for each of those reactions. So you can see with this core 1, it worked very well and most of the reactions went to completion. Core 2 didn't work so well, and then core 3 and 4 did pretty well, and most of the reactions worked okay for those. So what we did was we tested at a single concentration those reaction mixtures in the spectral shift assay. So just as an example here, what we were looking for was both good binding and also a good product purity, so reactions that had gone to completion ideally. So you can see here for some of the core 1 hits, you got this is the binding signal here, and we've got quite a good binding signal in most cases, and also most of the reaction mixtures went to completion. Whereas with core 2, there were fewer hits, and also most of the reaction mixtures were at a lower level. We did also we noticed a few potentially false positives. So these are where the reaction mixture doesn't seem to have worked, but we are getting a binding signal. So it might be interesting to actually test the building blocks for these samples, which we've not done yet, to see whether it's actually the building block is actually binding to the A2A receptor. So we then took the best compounds from that screen, and we resynthesized 22 of those hits that gave us the best response that we'd seen from the single concentration testing. So this is an example from the series 1. So compound 1 at the top here is the original fragment hit. So the crude sample gave an affinity around about 16.5 micromolar. When we repurified it, we got an affinity about 48 micromolar. So that's a good ligand efficiency, and that's kind of the baseline for what we were looking for. But compound 2 here, you can see the crude sample, we thought that the affinity was going to be about 2 micromolar, but when we synthesized it, it was actually better. And that's maybe because the conversion rate for that particular crude sample was a bit lower than some of the others. But we've basically been able to demonstrate that we've increased the affinity via the D2B approach 200-fold. And we've actually increased the ligand efficiency of that original fragment hit via applying this method. So we've identified novel compounds and also improved affinity and ligand efficiencies by doing this direct biology approach. So in summary, we tested 148 synthesis reactions on 4 fragment hit cores. This took 1 FTE day of resource, and this would be compared to, it would take about 7 weeks, we estimate, to use traditional synthesis and purification methods to do the same thing. From this, 21 fragment hits were identified with measurable affinities. 18 out of 21 of those reconfirmed after we synthesized those hits, and we managed to obtain up to 200-fold improvement in affinity by this direct-to-biology approach. So now that we've validated this method for the A2A receptor, we could now go back to it and actually apply larger D2B libraries, because we only did 148 in this case, but we normally do at least 1,000. So altogether, to summarize, there's no one biophysics technique that fits all. It's very much target and application dependent. We've applied all these biophysical techniques to a wide range of challenging targets throughout the drug discovery pipeline. And the combination of novel assays, hit finding, and D2B chemistry can accelerate drug discovery projects compared to more traditional approaches. And finally, just to acknowledge my colleagues, or at least some of them. So in the bottom right picture are all the people who do the biophysics work. So I'd like to thank all those people listed on the left, and also other colleagues in both protein sciences who help make the proteins and purify them, and also our colleagues in chemistry for the D2B work that they did. I'd like to thank you, and any questions?
Excellent. Thank you very much, Philip. So yes, now's the time. If you have any questions, please feel free to use the chat box, or I will keep my eye on the participant list if anyone prefers to raise their hand instead. Either is absolutely fine. And whilst we're waiting, so thank you for those very interesting case studies, Philip. You presented quite a wide variety of techniques. Does DomainX plan to expand those capabilities further in terms of different techniques?
Yes, we definitely like to. There are some techniques that we can currently subcontract, so we'd like to do more NMR work. There are a couple of different places that we can go to for NMR, so that's definitely something that we're looking into. There's also a number of other techniques out there that we'd like to be able to use, but it's just a case of prioritizing those over other, capital investments that we need to make. So yeah, there's definitely other things out there that we'd like to bring into the biophysics techniques that we can use.
Thank you. And is there a technique which is, so when it comes to very challenging targets or notorious ones, is there one in particular that seems to stand out in terms of being suitable for those challenging targets?
It's very difficult to pick like one technique. What we do when we're talking to clients about projects is just to get as much information as we can. So we will look at the target, things like the size of the protein, whether it forms dimers or higher order oligomers, what tool compounds are available. what likely expression level you can get and how much protein you have to be able to use, because if expression levels are difficult, then you're probably unlikely to be able to use ITC, and techniques like NMR might be difficult as well. And if it's a very big protein, that may make GCI more difficult. So we usually look at all those different bits of information to decide which would be the best approach for that particular target. I don't think it's not easy to say that there's one technique that would be best.
So there's a lot of factors at play there, I understand.
Yeah, exactly. Yeah.
Thank you. And in the case study you presented for the A2A, how long did it take to run the fragment screen for the A2A receptor?
It took about 2 days to run the fragment screen, and then it was also to do the initial concentration responses of the hits from that was also another couple of days.
Okay, I'm just keeping my eye out in case if anybody chooses to raise their hand or drop a question into the chat box. We've still got some time, so it's your opportunity to ask questions directly to the expert. So this could help you with a current challenge that you're currently facing. Which technique do you use most often, Phil?
GCI is the technique probably that we use the most. Yeah, we've got 3 GCI instruments at DomainX, and they're pretty much all used every day at the moment. Spectral shift, though, has been gaining in momentum, and we definitely get a lot more projects these days for spectral shift than we used to. And I think that the kind of upgrade from MST and TRIC towards spectral shift has made a big difference. We definitely get a lot more projects that we can use spectral shift for now than we used to with the other techniques.
Yeah, I was going to sort of ask, actually, sort of given trends overall in terms of what the industry sees in drug discovery, how is that guiding sort of the projects and the work that is coming away? Are you seeing any changes, or is it sort of not so much?
Yeah, I mean the change is really to get more challenging targets. I mean, things like membrane proteins obviously challenging, and things, there's a lot of projects that come to us that are protein complexes, and intrinsically disordered proteins are another example where they're typically quite challenging targets to work on. So we are getting a lot of those type of projects.
Thank you. And on the sort of sticking with the spectral shift, so what does it mean if the spectral shift signal is going in different directions?
It's a good question. So the information that Nanotemper give is that it's based on the hydrophobic environment of the fluorescent dye. So a downward change in binding is a decrease in the hydrophobic environment of the dye, and an increase in the signal is a more hydrophobic environment. So for an example, most of the protein-protein interaction experiments that I showed, you're seeing an upward trend. And so 2 proteins coming together is more hydrophobic. So that correlates with that kind of hydrophobic environment. What it actually means can be more difficult to say. It could be that if you're seeing different directions for different compounds, it may be they're binding to different sites, or it could be the compound itself is more or less hydrophobic. So it does provide useful information, but further work is required to try to understand it.
I see. Yeah. Thank you. Are there specific fluorophore wavelengths that are required for the spectral shift system?
Yes. So Nanotemper don't say exactly what their dye is that they use for they provide the labeling kits for the experiments. The emission wavelength is 650 and 670 nanometers. So as long as the dye is compatible with that, so dyes such as Sci5 are quite compatible with spectral shift. So it's similar fluorescent dyes to Sci5 is kind of what you're looking for.
Thank you. And do you always use biophysical techniques for D2B?
No, we also we can apply direct topology to cell-based assays as well. And there are other assay types that we can use, but it's mostly been biophysics and cell-based assays so far.
Excellent, thank you so much. Okay, still a few minutes left if anybody has any burning questions. As I said, now's your chance. And then we will be disseminating the recording post-webinar as well. That will come through from DomainX. So if you want to recap, you can, and then I'm sure they won't mind if you have any follow-up questions that you'd like to get in touch with. I'm sure that's no issue at all. And then the fourth installment of the webinar series is taking place on the 9th of December, and that's on the topic of covalent compounds. I've got one further question here. What would be your wish list for development of new technologies? I like that one. What would you like for Christmas? What's your wish?
Oh, this is such a long list. I mean, we would like to have access to NMR. There's a technique that's now available from Bruker used to be dynamic biosensors, that we'd like to be able to use. We'd also like to do more mass spec type of biophysical work. So things like native mass spec and HGX-MS would be techniques that we'd like to be able to do. But all these techniques require a lot of investment, so cost a lot of money. So at the moment it's difficult, but we're going to try to get access to those.
Yes, we're in an expensive business, aren't we?
Yeah, exactly.
Okay. Okay. Well, if there's no further questions, you'll have 5 more minutes of your day back. But I mean, I found that really, really interesting, Phil. Thank you so much for the time you've taken to present today. And then for everyone online, thank you so much for joining us and for all of your questions. If you'd like to register for the 4th installment of the webinar series, there will be a link going out in the follow-up email that goes around, so keep a lookout for that. It's also currently on the One Nucleus website, so you can register by going to our events page and looking for it there. So yeah, we hope to see you on the next installment. Thank you so much again. Thank you, everyone, and take care. Bye.
Thank you. Bye.